This is a course in next-generation sequence analysis, and at the beginning of the course, we want to introduce what the sequencing platforms are and the different sequencing technologies. I also want to introduce what we mean by next-generation sequencing. The second thing I want to do in the upcoming segments is discuss how the technologies themselves introduce certain sequencing artifacts, which is a major consideration in next-generation sequence analysis. We need to be aware of the types of artifacts that the different sequencing technologies introduce into the data, and later in the course we'll discuss how to correct for those artifacts. So before going on, I'd like to introduce a little bit of historical context just to understand when next-generation sequencing appeared and distinguish it from the historical way of sequencing. So before 2006, we used what we call Sanger sequencing. This is widely used in molecular genetics and many other areas of genetics, where we simply needed to know the sequence of a short molecule of DNA. In 2006, technologies we referred to at the time as massively parallel sequencing emerged, and these technologies we now refer to as both next-generation sequencing, but also a subclass of next-generation sequencing, that is, second-generation class of sequencers. We distinguish second-generation sequencers from third-generation sequencers, which are long-read sequencers. Okay, so second-generation sequencing is sequencing of short sequences of DNA in massively parallel context, whereas long-read sequencers, the target is to sequence much longer fragments of DNA. So during the next few minutes, we'll be discussing many of the most important sequencing technologies, including Illumina sequencing by synthesis, ion torrent semiconductor sequencing, and for third-generation sequencers, we'll be discussing PacBio, single molecule real-time sequencing, and Oxford Nanopore sequencing. I'd also thought I'd just quickly mention several of the technologies that have only gone defunct in the last few years. So it's 2019 now, and these technologies disappeared roughly 2015 and 2016. So the Roche solid sequencer you may see in the literature, you may even analyze a data set with that type of data at some point, but those are no longer being produced. 454 Life Sciences technology was another technology that was prominent up till around 2012 and 2013. For long-read sequencing, there's really only been one technology that disappeared, and that is the Helicos single molecule sequencing. Now, there's two technologies that have just appeared in the last 6 months to a year, and that's a Bio-Rad New Bio. Actually, this technology has not yet been released. There's been press releases about it, but the technology has not formally been released yet. QIAGEN Gene Reader, this is a clinical sequencing machine meant to deliver end-to-end sequencing in a clinical context. And finally, BGI Complete Genomics is a very new technology that may at some point attempt to compete with Illumina and the type of sequencing that Illumina delivers. And so we'll be talking more about Illumina in the next few minutes, as well as some of these other very important technologies.